In this video, we will discuss about this TEA LNPs results on resting and activated T cells. The reason this comes into the mind of researchers is might be because of the difference in the metabolism in resting and activated T cells. So they did the similar approach, that is, they took the T cells from the healthy donor and then divide them into different subpools, and a portion of the T cells is treated with the beads which has the CD3 or CD28 on their surface, and these beads are activated for 3 days before they are co-cultured with the T cells, a portion of T cells. And in the other case, what they did is they activate the beads which has the CD3 and CD28, and 11 days after activations, it is assumed that the beads might have gone into the resting state, they co-culture them with the T cells as a control. And in this case, once the activated T cells and the resting T cells populations are ready, they co-culture them with the LNPs, and the mRNAs that they used is similar to what they have used in the previous studies, that is sci-fi labeled eGFP mRNA. So the amount of sci-fi that are in the cells will gives us the indications about their uptake of the LNPs, and the eGFP signals gives us the indications about their translation efficiencies. And then they analyze the results using the flow cytometer. So let us go through the results in the panel B. For the resting cells, results are shown in the top row, and the activated cells results are shown in the bottom row. We can see that compared to the control, in all the cases, both in the resting and the activated cases, they are able to see around 90% of the signals, more than 90%, or approximately 90% of the cells have these sci-fi positive signals, indicating the uptake of the LNPs is efficient. Thereafter, the translation efficiency, the eGFP signals, is on the y-axis. They can see that compared to the control, the signals in the y-axis or the eGFP signals is significantly higher in all the cases. However, when they compare with the resting T cells and the activated T cells of the same TEA LNPs, they can see that activated T cells signals are significantly higher compared to the resting T cells for the eGFP expressions for all the 3 cases. And once they have this estimation, or once they get the idea that the activated T cells gives the better translation, then they also quantified the results to confirm their observations. So in the panel C, we can see that for the case of control, they are not able to see any significant changes for the untreated case, and but when they check it for this different TEA LNPs, they are able to see the percentage of this left hand side of the panel shows the sci-fi quantifications. So you can see that sci-fi quantifications are high compared to the control in the donor 1, donor 2, and donor 3, and this indicates that the uptake of the LNPs is efficient. Then they compare the results, or the quantify the results of the expression of mRNA using eGFP signals, and we can see that for the activation case, the signals are significantly higher compared to the non-activated case for all the 3 donor. And one more thing that they have observed, that the signals for the TEA41 that is TEA41 is lower than TE13 and TE37, and this is also in line with their dose dependent studies observations. This indicates their reproducibility of their method that they have used. And the heat map plot is also shown in the bottom of this panel C, where we can see that for the case of activated cells, the sci-fi signals intensities is high compared to their counterpart, that is in resting cells. And similarly, for the case of here, the results are in the horizontal rows, that is TEA13 results in the horizontal, for then 37, then 41, and then we can see that the intensities for TEA13 and 47 is higher than TEA41. And similar results they have seen for the mRNA expressions. This mRNA expressions is higher for the TEA 13 and 47 compared to the TEA 41. We can compare it with this legend that is on the left hand side of both the plot. So I hope you understood how they check or how they validate the TEA LNPs on resting and activated T cells, and they found out that activated T cells has better translation compared to the resting T cells. However, uptake of the T cells seems to be same or almost similar, both the activated and resting T cells. So this is what they have concluded in this figure. I hope you understood the results that are presented in this figure and their reasoning behind this experiment. If you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.